Hi all, in this video I'll be talking about the whole process of Sanger sequencing method. Okay, so first we will start with what is the reaction mixture the sequencing method needs. Okay, the first we will start with, or the first thing that should be started with, the synthesis of the complementary strands of that single stranded DNA which should be sequenced. Okay, so first we have to polymerize the complementary strands. So now you need the mixture. Okay, so what is the mixture? The mixture contain in Sanger sequencing method, first thing obviously a suitable primer. What is the primer? Primer is obviously the sequence which will bind to the DNA and help to initiate the DNA replication. So you need a suitable primer. Then you need clenofragment of DNA polymerase 1. Okay, I have already mentioned and discussed about the clenofragments function in my previous short video. So if you want, you can go and check my channel. Number 3, that is obviously mixture of DNTPs, means DATP, DGTP, DCTP, DTTP. Okay, this will be needed because these nucleotides will be added to the growing 3' end of the complementary strands of the DNA. Okay, number 4, that is very important, that I'll come further in the following. At least one of the DNTPs should be radio leveled in this case, as we have to trace or track that which sequence or which nucleotide has been added. So at least one of the DNTP has to be P32 leveled. Okay, number 5, a very small amount of dideoxynucleotide triphosphate, means DDNTPs. What is the main difference of DNTP and DDNTPs? DDNTPs that will lack 3' OH group, but DNTP, which is the DNTP that has the 3' OH group. Okay, but in case of DDNTP, which is basically the dideoxy nucleoside triphosphates, that doesn't have any kind of a 3' OH. So whenever DNTPs are present in a reaction mixture, by chance if they are incorporated in a growing DNA strand, suppose you are adding DATP, DGTP, something like that, if there is DDATP by chance added at the end after any kind of a normal DNTP, at that place, at that condition, the replication will be stopped. Why? Because the clenofragment doesn't read or doesn't identify it is a specific it is a different nucleotide from this one. So it's a very unknown thing for clenofragment of the DNA polymerase 1. So immediately it will stop its function, and at that place the whole DNA polymerase reaction will be stopped. So we will get the truncated ends. Okay, we will get the truncated ends, but have the same 5' end. Why? Because we already selected clenofragment. What is the function of clenofragment? Because as it has 3' to 5' exonuclease activity, so we will get the 5' exact, all the same 5' containing DNA fragments. But as we also have added DDNTPs, the clenofragment, they will identify that it is not my type of nucleotide that I should further move on, so it will stop working and DNA replication process will be stopped at that position. Okay, so in this way we will get the fragmented DNA. Now what will happen in case of fragmented DNA? There is 4 reaction mixture. One, that is obviously with DATP plus DDNTPs plus primer plus Kleno fragment, okay, plus the DNA template. This is the reaction mixture 1. In reaction mixture 2, there will be DGTP, and all these things are similar. In second one, this one is the second. Third one is obviously DTTP, and rest of the components are same. And in case of DCTP, rest of the components are same. So in this way, 4 reaction mixture will be prepared, and in a different reaction vessels these fragments will be produced, and then we will have to run them in sequencing gel. I'm really sorry, but my markers are not properly working, so please follow my instructions, okay? So suppose the first reaction mix, you will get only DATP containing fragments, okay, that will be DATP containing fragments. The second thing, that will be DGTP containing fragments. The third one is DCTP containing fragments, and fourth one is DTTP containing fragments. Now this is a sequencing gel, okay. If this is a sequencing gel, there is A, G, C, T, something like that. So from different reaction vessels, you will pick up and put your reaction mixture into the sequencing gel well, okay, the wells. Further in the RDT classes, you will learn about it, how you have to prepare the gel, not the sequencing gel, but how you will prepare the gel for gel run, okay, after PCR or something like that, the DNA fragmentation and all. So in this way we will learn about it, how to prepare the running gel buffer and all, and what is the well I'm telling here. So after this, suppose you will get a band here. This is A, okay. You will get a band here. For G, you will get a band here. Suppose all of these are arbitrary. You will get a G here. Then you will get a C here, and there will be, you will get the T here. Again you will get A here, then G here, then you will get C here, okay. The lane at which the wells are made, you will put your samples into that, and you will get the gel bands accordingly. It's not like you will get the bands of C at the lane of G. No, it's not like that. You will get the bands of C according to its lane, according to its lane. They will not be mix matched. So that is the beauty of gel run or gel electrophoresis. So after getting that, what is this? This is A. So write it down. This is the process, how you will know the sequence. What is the sequence you will get? So first you will get A. So this is A. Then second one I have told you, that is G. You will get G. Third one you will get C. Okay. Fourth one is T. Then again, what is the line? This one, G. Again you will get G. Then again A. Okay. Then last one is C. So this is a sequence. Now this is how to read this sequence. What is the process of read the sequence you will get at your gel? You will just read the sequence from bottom to top, not top to bottom. You just have to read from end to start. So in general thing, you will start like this: A G C G A C. No, it's not like that. You will have to always remember that you will read the sequence from bottom to top. So what will be the sequence now? C A G C G A. Oh, sorry, there is a T also. I've missed. So this is the sequence of the DNA you want to sequenced. Okay, and you will get it from the complementary one. As the sequences are complementary one, these sequences are the complementary one of the sequence of interest or the DNA of interest. So you have just reversed it down. Okay, you will have always read out from bottom to top of the gel. Okay, from the bottom to top of the gel, you will have to read it out and write it down in this way. So we will get the sequence. So this is the easiest way I am trying to make you guys understand, but this is not so easy. There is a computer generated softwares and all who will read it after all. So in this way Sanger sequencing process works. Thank you so much. If you like, please subscribe my channel.